We can see now, finally, the regulatory T cells, the inducible type that produces FOXP3. And then as you notice here, in terms of cytokines produced, then there's this array, again, CD8 and CD4. Then if we're looking at CD4, we see that there is indeed a stronger response to antigen and light map in experimentally infected animals. If we look at naturally infected, the pattern looks fairly similar. But the important point to be made here, in contrast to previous reports, what we're seeing is the proliferative response is not diminished at the clinical stage of infection. And the dogma has been that the cell-mediated immunity is diminished. So this is a contrasting bit of information that we have obtained. Then we look at CD8, we find in the experimentally infected animals that there's a poor response to soluble antigens early on, but there is a dramatic response to light map that we haven't been seeing in the past. And if we look at naturally infected animals, then we see that there is a response to soluble antigens and to live map, and a stronger response still to live map. Now, if we're looking at FOXP3, we've finally got the antibodies that we developed to work. So we believe we're really seeing FOXP3 positive cells. So if we look at the cells unseparated, so we're looking at memory cells going up, and these are CD25 at the same time, but we're not showing that. But we can see that we have a positive population of blast cells expressing FOXP3. If we split it out so we can see this in greater detail, then we can see that the activated cells do not express FOXP3 here, but over 40% in the clinical animal are expressing FOXP3. So we see that there's active events transpiring in a culture and things we haven't been able to see before. So then I have limited the cytokines that I'm presenting here, but we're trying to look at a larger set and trying to make some sense out of what we're seeing. So if we're looking at experimental and naturally affected gamma interferon, in this study with these animals, the gamma interferon is a little higher in the clinical animal. If we're looking at IL-17, it doesn't really look that much different, but it is upregulated over uninfected animals. If we look at IL-22, then in this study, it's higher in experimentally infected animals than naturally. And both of these cytokines, under normal circumstances, they're protective. Under dysregulation circumstances, then they become inducers of inflammation. So we can't tell one from the other. We can see at this moment that they're present, and importantly, it's saying those cell populations are present in preparations of ileal cells taken from the intestine. If we look at FOXP3, it is up, but it's up early and it's up late. So it could be playing a regulatory role that's protective early on, and then it could be disruptive at a later stage of disease, which isn't revealed here. Then if we look at granulysin, surprisingly, my initial hypothesis would be that at the late stage of infection, that part of the pathology would be that there would be downregulation in the expression of the granulysin gene, and this is not the case. So the story is obviously very complex. Then if we look at the endoscopic evaluation at the early and late stages of disease, then what we're finding now is that serial biopsies show MAP isolates from humans and cattle persist at a low level in the ileum without development of lesions up to 15 months post-infection. But MAP is readily detected in biopsies from cows at the clinical stage of infection, which we'd expect since all of the histology and immunohistochemistry can demonstrate the bacteria easily also. So in summary, where we are, pathogenesis of CD is associated with dysregulation of several arms of the immune system. These are the same pathways that are targeted for MTB, MDB, and MAP. So that this implies that MAP could play a role in pathogenesis, even if it's not the true etiologic agent. It is a pathogen that clearly can cause disease. So we believe now that the bovine cannulated ileum model provides an opportunity to identify the trigger that dysregulates the immune system in Johne's disease and potentially chronic or Crohn's disease. And I think this is what we're really after, is to get a model system where we can dissect the immune system and the events of dysregulation. And this could be several points in a process of this sort, so that it could be similar, but the actual trigger could just be in the same pathway. And under these circumstances, I think we have a chance to work with this because it's a live model, and we don't have to kill an animal every time we want to do a study. We can do a sequential study that should be much more informative. So with this, these are the groups that have supported the work up to this point. Thank you. These CD8 cells that we were looking at are, we're gating on those when we obtained this information, so we excluded the CD8 positive NK cells and the CD8 positive gamma delta cells. So that this is where it's important in the model system to use electronic gating to isolate the population that you're really interested, to exclude the possibility that you're not looking at what you think you're looking at. Very Pardon? We have two antibodies. One is a IgG 2B and the other is IG1. So that we've looked at these, I'm forgetting which one we used in this particular study, but we've looked at these in parallel with isotype controls to make sure that we're actually seeing it. And then what's important that we hadn't appreciated is in the bovine system, the buffer reagent used to get the antibody to work is critical. And eBiosciences happens to be a company that has put something special into their cocktail, because we've done BD and we had our own cocktail and we were not successful. So this is was really a breakthrough.
Do you think there are very many more cell types to be identified and cytokines to come? Because I think a lot of people are having trouble coping with it. And should we have cytokines and cell types for dummies?
Well, this, I'm right at the forefront and I'm asking the same question. Why do they keep finding these extra things? Because I had a hypothesis that it explained everything without these subsets being present. And now they say here they are. And then the mouse literature is very good, but the Th17 is producing IL-17 and IL-22. And in humans, there's clear evidence that there's two subsets, and I suspect in cattle, this is going to be the same thing. And we can't exclude the possibility that there are additional subsets, which means we have to make more reagents.